my good friend, the Dean of the College of Science of UP Diliman, Dr. Cynthia Saloma, who is in fact a native of these parts. The others are keynote speakers, Dr. Menchi Lagman and Dr. Chuya Shinzato.
In the beautiful island of Boracay, this gathering of scientists, researchers and experts from all over the world is testament to the power of science as a unifying platform, a valuable reminder in the backdrop of a global landscape that seems to be increasingly fragmented and contested. Our hosting
everything since I got to the airport and meet the people and meet all the, all the transportation and getting into the boat, it reminds me a lot of South America and Colombia and Panama. I was joking with the people in, the port this morning that when we got to the port, it's almost like if you go to Bocas del Toro of Panama. There is all these people and all working, and there are all these bands.
Was actually, so that was 2021. Shortly afterwards, we had a second GIGA board. So every time we do a GIGA meeting, shortly after we have elections. So if anyone here is interested to run for any of these positions, please manifest yourself. We talk about it during the GIGA business meeting tomorrow, when you will know how you can participate in the association in any of these roles. And so the president in the second governing board was Todd Oatley, and Joe, new election. And now that's when Carlos became our president. And again, I came back with a desire for revenge to really make GIGA happen in the Philippines. I took again the role of vice president for conferences. And finally, we made it here. We're all here. So I'm pinching myself because it's like a dream. It's been planned for so many years, but yes. It's a great pleasure welcoming you all, like a dream come true. And I hope you'll have a wonderful time in Boracay, in the Philippines in general. I hope you'll be staying a little bit after. We put a 1 week between the ICRS and our meeting with the idea people could stay a bit longer in the Philippines before moving maybe to New Zealand.
The intersection of what we find in there based on their metagenomes and who dominates based on their metagenomes, we really see very, very big differences. And we're in the process of figuring out what's in the environment as well. And of course, the other side of this, which is functional annotation. It's easy to use ASVs for taxonomy, but they're not very good.
The bottleneck, and was thus classified as critically endangered. So after human intervention, and the population was able to recover in 2005, and between captive bred and wild captive heterozygosity levels, with the wild captive individuals having lower heterozygosity than the eagles that were born in captivity.
So we wanted to have more actually, but the problem is we don't have enough material for that individual. So we're kind of stuck with what we have and how to analyze this. So I've been looking for
how Filipino researchers work in line with doing research in the marine environment. So this spans basically not only invertebrates but also vertebrates here in the country. In marine and aquatic resources, so technically there are 2 agencies tasked.
Legal basis for conservation and protection of wildlife resources, their habitats, and funds and for other
Actually, Di ba tapos ang ating... Grabe na po. Cellphone. Grabe nga po, work. Hala, sige! Hala! Go, guys! Hala! Ay, sino siya na? Oh, kwayo?
So we will kick off today's session on genomics of marine holobionts, which is chaired by Dr. Cecilia Conaco of Marine Science Institute of the University of...
But we have also good coral reef. As you see, 400 coral species are reported. This number is almost comparable to Great Barrier Reef for Australia. And all of these coral reef in Okinawa. And this coral reef area in Okinawa is regarded as a marine biodiversity hotspot because many endemic species are found there. And of course, it's beautiful. Many tourists visit.
2 minutes. So we request everyone to settle in. Thank you.
Doses are not regained. Nevertheless, during a bleaching event, we might see that different species or forms of corals vary in how they tolerate heat stress. Some forms, like the massive...
This is all from one specimen, so we did both PCR Sanger sequencing here, right?
And that time has come. And so now we have openings for president. So if you want to become a president of GIGA, you can put your name, and then
Also, we have a student / early career postdoc position that could be on the board, which is not officially on the Invertebrate bylaws. But we've had always, at least for the past few terms, a student or early career researcher sitting on the board. So if anybody wants to volunteer on that, they are welcome to put their name in, contact us.
Say your name. Vice President of Development, which is the one in charge of filing money. Vice President of Early Career, which is what Joe just mentioned, that is usually a postdoc. And also a student, that could be, it's a student representative in the GIGA board.
That's different. So the Vice President of Early Career.
Always, he made the society possible, right? He was the David Burr. I think he's always been around.
Founder. Took a lot of people.
Some of us are since the beginning, but he really is the one who started it.
Connections between the phylogenetic signal and categorical traits, which can be really useful in a...
Contaminants, but you may ask. How do we know these are just not misidentification? Well, luckily through my SEM imaging, I was able to find a plethora of invertebrates and other cellulophores on top of the
Otago and Bioeconomy Science Institute in New Zealand. Today I'm going to talk about the research that our lab has been working on, specifically in developing high-quality genome assemblies to investigate the climate change resilience of key aquaculture species in New Zealand. Of what I'm going to present today, specifically in developing the green-loop mussel genome assembly, was actually done by my main supervisor, Nathan Kenny, while the other half on Chinook salmon was done by me. And so that for the case of green-loop mussel, it is known to be a Toonga species, meaning that it is also considered a treasured species by the Maori people, who are the indigenous people in New Zealand. And since they are considered as such, certain traditional practices called tikanga also have to be followed regarding their storage, processing, and disposal. Now for Toonga species, the management in New Zealand, in fact, it is considered to be one of the most successful fish transplants in the country, but at present
are different for each standard because they have different genome sizes. So I just normalize them by dividing each IOD by their known genome size. So they all should start with, like, the values should be more or less same, consistent. Then I will be able to merge them all in one column. The histogram to give you the genome size, but I found out was not accurate. So we use basically the mode of the kernel density, see the red line, and also was able to generate the coefficient of variation of the sample, so how accurate, how the data varies across the mean. And of course, this processes was.
Here we have now a panel on how to make it in academia. So it's a suggestion for student representatives in GIGA. And I think it's a very good idea because we are in a small group here. So Boracay researchers should feel free to ask any questions. We are among ourselves. If you wonder about some dirty little secrets of academic life.
Of a paper where public health workers work directly. And so looking at the overall, we did 3 calls of, we did 3 rounds of these data mobilization campaigns. We had really interesting
their genetic structure of the species. And so I hope to come back to GIGA. That was one of the main goals of coming here, was to participate and kind of get a little bit more background onto generating genomes. So part of this would be to generate that high quality reference, assess this genetic connectivity and regional stock structure using.
With the Judging crab is
they have quite a disjunctive distribution, because it's quite distribution. We have a cluster of species in the nootropics, but also we have some species recorded here, and they have quite a disjunctive distribution, because it's quite distribution. We have a cluster of species in the nootropics, but also we have some species recorded here, and
microbial resistance is recognized as a global health challenge. So therefore, there is an urgent need to identify the novel antimicrobial peptides, antimicrobial agents, we can say. So here the mollusk possesses the stronger innate immunity and rich source of you can see.
You know, if anyone would ever like to do any kind of a collaboration or has interest in students or anything, I'm always happy to help disperse the gospel of the different colors are different kinds of interactions, and the bottom line is the colors are not grouped. So like whether a ritualist or an antagonist or a commensal doesn't seem to.
The hypothesis and alternative hypothesis. The hypothesis, we have a single antipode that was abyssal, happy with his life, until he decided to went into Hadal and to Hadal depths. And then, one explanation is that he jumped across different Hadal trenches and colonized the whole planet. The other hypothesis is that we have an ancestral species that stay in abyssal stage and jump repeatedly into Hadal depths. So for the sake of simplicity of my project, I hope the alternative hypothesis is the real one. In reality, it's going to be a mix. In reality, it's going to be a mix. For example, we know already that these populations are really close, and this is probably a single event of colonization and then jump across nearby trenches. But
DNA-dependent RNA polymerase. So template is DNA, but RNA viruses use RNA as a template. So this is a much simpler enzyme than DDRP. So this is the enzyme, viral RDRP. So we can use that as a plate to recover RNA viral context from our transcriptomic data. And we need just to check if it has these active sites, motif A to motif C. Those RNA viruses contain those motifs, and we found around 8 high-confidence RNA viruses belonging to 4 orders and 1 family Microviridae. I'm showing you here one example. This is from Favides colmani, belonging to Picornaviralis order under this family right here.
An input coming in from there, and then we also have oceanic input coming in from here that is less nutrient rich, and this gives us a very sharp environmental gradient where we have very high levels of dissolved inorganic nitrogen in the water on the left-hand side and healthier waters on the right-hand side. And the coral I'm going to be working on is a copra, which is fast-growing and structurally complex, so it's great as an ecosystem engineer.
In the process, these events are recent and they are driven by segmental duplication and inversions, and we don't identify ancient Golden non-duplication events in copperpods. We think our hypothesis is that the genomic expansion is fueled by transposable elements and erroneous DNA repair, like in triggered by population isolation and by promoting inbreeding culture. It's important to mention that
Mabalik pa, sir.
compiles it and then provides a dashboard, an internet dashboard. And this is kind of how it works. Here, the genome sources, the raw data is there. The consortia provides a sequence, a species target sheet, and then there's some other records that go into the genome hubs, which is.
And then you have to also deal with these major undertakings. They had to organize, all these groups organize their scientists who want to collaborate and get their genomes read. And it requires expert identification, expert biology, forming this community, opportunities to create new collaborations, forge new approaches to access and share indigenous groups and local communities. So all this is just kind of a snapshot of what has been established already.
There are corals living in turbid zones. In fact, the corals, there are some reports which show that corals living in turbid zones are more tolerant to bleaching and they are more resilient to environmental stressors. So yes, this happens. Ours is not a unique place, but then which means that we are at the right place, right? Like corals that are under stress are more resilient, which means they could be future-proof.
Of course, I think the people who are working on this, they have to work on that, but then now we are looking after this facility. So IVF has been done. Around one pair is responsible for adding 7,000 to 8,000 juveniles into the population. But as the project says, there is a scope for future, and this also has to be addressed in the next 5 years. So we are talking about synthetic derivatives. We are talking We are talking about the
focusing much more in the last few years, which is understanding chromosome evolution. Okay, across different taxa, and then it's basically chromosome architecture, right? So we need complete genomes to understand how the repeats are there, the composition of other known.
On the top of the shell instead of a slit. And among the broader group to which they belong, which is called gastropods, they have very rearranged mitogenomes. So what we are seeing here, so everything that you see in white are the genes of the mitogenome, which are conserved in gene order compared to what's inferred to be the ancestral gene order for this larger group. And what we see here in red are the fusurelles. So the few mitogenomes that have been known so far have very rearranged.
Second award goes to Kelly Ron Publico.
Thank you and congratulations, guys. For the best poster presentation, may we
Presentation. This is my book on coral conservation and assessment biodiversity. So this goes to Jing Hui.
Models today, genome assembly and genomics, comparative genomics, and that has been wonderful. And I hope that has inspired you to do some different analysis that you want to do. You have also met a few people.
Antas ang agham dito Pilipinas. Good science, great science, stimulating work, very inspiring. So, yeah, I hope we have another one in the near future. We get it ready, and we look forward to having elections coming months. Just a new.
So it was wonderful having you all here. A dream come true. I told you the story before. We had both excellent science, some cultural activities. We had okay weather, not the last day, but I mean, until that point, it was all fine. So I think we rubbed our antennas, our tentacles with each other and exchanged a lot of ideas. Please raise your hand for South Africa. Okay. Raise your hand for Madagascar. I'm gonna need to count the hands afterward, but it's fine.
Same. Papagusto lang.
Raise your hand for Tanzania.
Tanzania.
Okay.
Ano siya sa
For Cape
Tanzania?
Verde. You have to count fingers. Azores.
Si Gonzalo eh.
Canary Islands. I think basically we're pretty excited to meet again. But wherever, you know? Okay. Yeah.
Kadamo-damo, New Zealand, Cebu.
Hey, Colombia. So returning to Colombia, but on the other side this time. And finally Saudi Arabia. So I'm gonna have fun counting the hands and fingers afterwards, but we get to rough idea.